He is a recipient of the 2013 Potamkin Prize for his extensive work in Alzheimer's research. His laboratory is widely known for using positron emission tomography and magnetic resonance imaging to understand brain aging and dementia. He's on the editorial board of several journals and leads the PET imaging component of the National Institute of Health's Alzheimer's Disease Neuroimaging Initiative. Now please join me in welcoming Dr. William Jagust.
Thanks for coming. Thanks for spending a beautiful Saturday indoors in a dark room. I'm going to talk to you about my research and Alzheimer's disease. And this is an area I know many of you are interested in neuroscience. And we tend to think of neuroscience in terms of disciplines. Some people use molecular approaches to study the brain. Some people use electrophysiological approaches. Some people use cognitive approaches. And my lab pretty much uses cognitive approaches, but we really also get into some of the molecular biology. So this is an area that I think brings together some of the molecular and cognitive and systems aspects of neuroscience to think about how the brain changes in aging. So let me give you an overview of what we're doing. So I want to start by defining terms. These are medical terms, and I'm going to use them a bit. So I want to make sure we're all on the same page. So dementia is a global and progressive loss of cognitive function that interferes with a person's ability to do the things they need to do during life. So a person with dementia obviously has trouble with things like memory, but they also have difficulty driving a car, finding their way around, getting dressed, taking care of themselves. And Alzheimer's disease is the most common cause of dementia, so they're not synonyms. Alzheimer's is kind of a subset of dementia. It's the most common cause. It begins usually with memory loss. And the kind of memory loss that people with Alzheimer's disease experience is the inability to form new memories. So an Alzheimer's disease patient can remember something that they learned a long time ago or something that happened to them decades ago. But if you talk to them and then come back a little while later and ask them if they remember what you talked about, they'll have forgotten it because they can't form new memories because the systems in their brain that are responsible for creating new memories are undergoing degeneration. Now, Alzheimer's disease is characterized by a pathology, and that's really what I'm going to spend most of my time talking about, are the actual pathological changes in the brain. And in Alzheimer's disease, there's two different kinds of pathological processes. One of them are called neuritic plaques, but we now more often call these amyloid plaques or beta amyloid plaques. And this is an amyloid beta plaque. It's a large conglomeration of protein that aggregates, it sticks together, and it forms this sticky substance in the brain called beta amyloid that many people believe is a very key event in the cause of the disease. The other pathological feature of Alzheimer's disease is tau, which is in the neurofibrillary tangle. And this is a neurofibrillary tangle. These are neurofibrillary tangles in neurons composed of this tau protein. Now, this is still one of the mysteries of Alzheimer's disease. We don't really understand how amyloid or A-beta and tau are related. We don't really understand how one might lead to another. We don't understand how either comes about and why they necessarily accumulate in the brains of many older people. And so really, a lot of what I'm going to tell you is a theory. It's a theory that people call the amyloid hypothesis. And it has a lot of problems with it, but I'm not going to tell you those today. I'm going to make it easy for you. So what I'm going to do is give you the dominant theory of Alzheimer's and the evidence for it. And maybe next year I'll tell you why it might be wrong. And actually, that's a good lesson. I know many of you are headed to medical school probably, and I always, I don't say this very often, but I remember there's a famous quote about medical school where the dean tells the class, you know, we realize that 50% of what we're teaching you today is going to turn out to be wrong. We just don't know which 50%. Okay, so here's the sort of molecular story about Alzheimer's in a nutshell. This amyloid protein, this A-beta, is shown here in white. It's a cleavage product of a much larger protein that's about 700 amino acids long and sits crossing the cell membrane, as you see here. And it's cleaved by this precursor protein called the amyloid precursor protein. is coded on chromosome 21, and it's cleaved by two enzymes, a beta secretase and a gamma secretase. And when those two enzymes work on this precursor protein, this A beta is freed. It's soluble in the brain, but then it aggregates to form this plaque. So it exists in this soluble form and then in this aggregated form, which looks like the plaques we're talking about. So that's about a decade of work in a minute. Not in my lab. Other people have elucidated this. But the reason it's so important is because it's been the basis for a lot of thinking about the disease and also for treatments. So if you look at this, you can imagine there are ways we could potentially interfere with this process, right? So one of them now that's gained a lot of enthusiasm is the idea of using antibodies against this amyloid that could suck the amyloid out of the brain and lower it. The other part of the story is enzymes that might be inhibited. So if we could block these secretase enzymes, we might inhibit the cleavage from happening, and we'll have less of this A beta. Now, of course, why would we want to do that? And that's where the story really comes in about the amyloid hypothesis. And most of the evidence for the amyloid hypothesis, or at least the strongest evidence for the amyloid hypothesis that's been around the longest, are genetics. So Alzheimer's disease is very, very rarely caused by genetic mutations. When I say very, very rarely, it's hard to know how many cases. It's way under 1% of all Alzheimer's disease. So it's very rare, but in some families, there's a gene that's transmitted from generation to generation that's autosomal dominant, and there's 3 different mutations that are carried along that could cause Alzheimer's disease. Any one of these 3 could cause Alzheimer's disease. What's very interesting about these mutations is that they all affect the processing in some way of this precursor protein. So one family of mutations is in this amyloid precursor protein gene. So people actually have mutations that code for this protein on chromosome 21, and the individuals who have these mutations get Alzheimer's disease. Now, one other thing about these mutations, in addition to being extremely rare, they're also very unusual. Sometimes they don't look like typical Alzheimer's, but the most unusual thing is their age of onset. People with these mutations usually get the disease in their 30s, 40s, or 50s. And as I'm sure you know, most Alzheimer's disease is the disease of people in their 70s and 80s. So it's very unusual. But in any case, individuals who have these mutations in the precursor protein will get Alzheimer's if they live to be 30 or 40 or however old the disease is in their family. So that's one group of mutations. Another couple of mutations occurs in 2 proteins called PS1, that stands for presenilin 1 and presenilin 2. And these proteins are part of this gamma secretase. So this gamma secretase enzyme, which cleaves the precursor protein, could contain a mutation in its subunits that also cause Alzheimer's disease. Now, what's interesting, so you're now seeing, okay, this is an interesting link between these genes and Alzheimer's disease, but what's the most important is that all 3 of these mutations are associated with altered processing and increase the amount of A beta that's produced, or if they don't actually increase the amount of A beta, they increase the amount of one species of A beta that's called A beta 42. It's a 42 amino acid length protein, because that A beta 42 is probably the most toxic form of amyloid. It aggregates more potently and it affects neurons. So all 3 of these autosomal dominant mutations affect amyloid processing, increase the amount of amyloid or A beta 42, and cause Alzheimer's disease. There's a 4th gene, which isn't a causal gene. It's called apolipoprotein E, and it's a polymorphism. That means it can occur in different forms. There's no form that's normal or abnormal. It occurs in the form of ApoE2, ApoE3, and ApoE4. And if you have ApoE4, you're at an increased risk of Alzheimer's disease. And we also know that ApoE4 is associated with more amyloid deposition in the brain and reduced clearance of amyloid out of the brain. So in some of these genetic causes, it seems there's overproduction of the amyloid. In the ApoE4 situation, it seems there's underclearance of the amyloid. So this has all been really, really strong evidence for the amyloid hypothesis of Alzheimer's disease. And it goes just like this. All the autosomal dominant forms of amyloid affect amyloid precursor protein processing in some way, and they increase A beta deposition. Now, I didn't talk about the other genetic cause, which is Down syndrome. I don't know, many of you may know, but some of you may not know that children who have Down syndrome who live into their 30s, 40s, and 50s actually get Alzheimer's disease. They have these plaques and tangles in the brain, and for all intents and purposes, they seem to develop Alzheimer's. And if you think about why that might be, they have 3 copies of chromosome 21. Chromosome 21 codes the amyloid precursor protein, so they produce more of this A beta as well. So that's the explanation for Down syndrome's association with Alzheimer's disease. So another interesting genetic cause. As I said, the most common genetic risk factor is the polymorphic ApoE, and that's associated with increased deposition of A beta as well. So now one of the major laboratory methods of studying Alzheimer's disease is the production of transgenic animals that have these genetic mutations placed into them. And these gene mutations can produce behavioral deficits in animals as well. Well, A beta is the hallmark of the diagnosis of Alzheimer's. So if we see those plaques in the brain, then we diagnose Alzheimer's. So that's another key bit of evidence. But if you think about that for more than about 30 seconds, I hope you realize quickly that's really circular logic, right? So I'm not sure that's the greatest piece of evidence. But the next bit is kind of interesting, and that is this amyloid, this A beta, when it occurs, especially in these soluble forms, seems to affect synaptic function. It affects both the electrophysiology of the synapse and it affects the structure of the synapse in bad ways. And so there's evidence, at least in some situations, that A beta is toxic. So this is pretty good evidence to me, and it's evidence that any theory of Alzheimer's has to deal with, that the idea is that overproduction or underclearance of A beta is the cause of Alzheimer's disease. Now that's a very bold statement. And it's not completely subscribed to. And there's a lot of problems with it. And here's a few of the big challenges to this. And these are the big things I'm going to deal with today. There are a lot of other issues, but here are the big challenges. The first big challenge is that there's a poor relationship between the amount of amyloid in the brain and what a person's cognitive symptoms are. And the best example of that is my next point, which is that older, cognitively normal people can show a lot of this A beta in the brain. So you can be completely normal at the age of 75 and have a lot of this A beta in the brain and not have any symptoms. And then finally, there have been a number of clinical trials of drugs that actually lower A beta in the brain, but don't improve the health of patients, don't make them better. So these are 3 things that I'm going to talk to you about, and I'm going to show you evidence for why I think these aren't really such important challenges to the amyloid hypothesis. There's evidence for why all of this could still be consistent with the idea that A beta is a crucial cause of Alzheimer's disease. Let me talk a little bit about the methods we use and a little bit about what we've seen in patients with Alzheimer's with some of these methods. So I look at this from a historical perspective. I've been doing this a while. And the first way we were able to really look at the brain in the modern era is to look at its structure. So with things beginning with CT scans and then more recently with MRI, we can look at the shrinkage of the brain or atrophy using MRI. Beginning in the 1980s, people began to apply PET scanning, and we could see changes in glucose metabolism. And this gives us an idea of steady state neural activity, what's happening in the brain in a sort of a steady state situation in terms of metabolism and brain activity. Beginning in the 90s, people began applying a functional MRI to look at brain activity during tasks and also networks in the brain. And I'm going to show you a little bit of work using that. And then most recently, we've been doing what people refer to as molecular imaging, using PET to actually visualize the proteins in the brain that are becoming deposited there. And so we really use these multimodal approaches in order to try to understand what's happening in aging. So here's the story with brain atrophy. We've been known for decades that the brain shrinks in aging, but it shrinks much more in Alzheimer's disease. And these are pictures of surface pictures of MRs of patients, a normal older person, a person with mild cognitive impairment, which for today we can consider sort of an intermediate stage between normal aging and Alzheimer's disease. These are people who are beginning to experience memory loss, but don't have the full-blown picture of Alzheimer's disease. And this is a patient with Alzheimer's disease. And you see, if you just look at the brain itself, you see there's shrinkage of the brain, there's widening of the sulci, there's shrinking of the gyri, even as early as mild cognitive impairment, and it's very dramatic by the time a person has Alzheimer's disease. But there's another interesting finding when you look at the brain in cross-section and you begin to quantify the structures in the temporal lobes. It's actually in the hippocampus, which these arrows are pointing to, that you can begin to see really dramatic shrinkage, also beginning very early, probably even in people who are normal, as I'll show you. We think the reason for this is that the hippocampus is particularly affected by these neurofibrillary tangles, by this tau pathology. And for those of you who have been, studying the brain and cognition, you know that the hippocampus is very important for the formation of new memories, exactly the kind of problems that patients with Alzheimer's disease have. So the idea is that the shrinkage of this structure related to the pathology of Alzheimer's disease is one of the underlying mechanisms for the memory loss in people with Alzheimer's. And we can look at all of this during life with MRI. With PET scanning, we see something rather different, and that is changes in glucose metabolism in the cerebral cortex. And it tends to show up in these rather characteristic locations that you see here in blue. So these are PET scans where the blue shows areas where glucose metabolism is going down. And the areas that are involved are in the parietal lobe, the temporal lobe, and the medial parietal lobe here in the precuneus and posterior cingulate. So you're going to see images that show this pattern now for the rest of the lecture. So I want you to get a good look at it because it's not a diagnostic test for Alzheimer's disease, but it's a pattern of dysfunction that reflects something that's very interesting, both in terms of the type of networks that seem to be involved in Alzheimer's disease. So this is the area of the brain that, again, in Alzheimer's disease becomes hypometabolic and glucose metabolism going down. Now, PET scanning is a very interesting technology because what we do with PET scanning is we basically inject people with a radioactive molecule. It sounds kind of scary, but it's actually, the scanner is so sensitive that we only have to inject people with really low doses of radioactivity, and we can see images in the brain of where this radiation is coming from, so we can map all kinds of biological processes. And one of the things that we are beginning to be able to do is to not just look at things like glucose metabolism, we can also look at neurotransmitters like dopamine, but we can also look now at some of these proteins. And I think the big breakthrough in this area was the development of this compound that was called Pittsburgh Compound B, which we now nickname PIB for short. Guess where it was invented? Okay. So a couple of guys, scientists in Pittsburgh, worked on this idea for a long time. And what they did is conceptually very simple. Technically, it's quite complicated, but conceptually it was very simple. They started with this dye called thioflavin. And if you use this thioflavin dye in a slide, on a microscope slide of a brain of Alzheimer's patients, you see that it binds to the amyloid plaque. And actually, it's not really clear how it's binding to the amyloid plaque, but dyes like this have been used for a long time to visualize the amyloid in tissue slices at autopsies. So you could see the amyloid pretty well with this thioflavin. So it seems like a pretty simple idea to take this molecule and label it with something that's radioactive so that we could see it in a PET scan. The problem is it's not that simple because this molecule here has a positive charge, and positively charged molecules don't cross the blood-brain barrier. So what these guys did is they worked for a while to try to modify this molecule so it would no longer be charged. They radio-labeled it with a positron-emitting isotope called carbon-11, and when they did that, they showed they could get it into the brain, and the amount of binding in the brain was proportional to the amount of amyloid in the brain. So this red color here shows you where this PIB is binding. And the redder it is, the more of the amyloid there is in the brain. So two things about this that are interesting. One is that the carbon-11 has a 20-minute half-life. So it emits radioactivity. We can detect it with a PET scanner, but you have to make it and use it very quickly. So we can do this here in Berkeley because we have a cyclotron up at Lawrence Berkeley Lab, and that's the way that we do these experiments. So as you know, last time you went to your doctor, he didn't open up his cyclotron. So this has been something that's fairly limited in terms of clinical applications, but there's now ways to do this using longer-lived radioisotopes. So everything I'm telling you today is actually possible on a much larger scale. The other thing is that this PIB binds to the fibrillar forms of amyloid, the kind that are stuck together in the plaque. And I mentioned earlier that there are these soluble forms that float around, and we don't see those with this approach. So these kinds of imaging things that I've been talking to you about, we now think of as biomarkers. They're biomarkers for different things that are going wrong in Alzheimer's disease. And in one of the more interesting projects that's been going on, these biomarkers have been applied to people with these autosomal dominant mutations that I've been talking to you about. Now, this is an example of data from this particular study. It's called the Dominantly Inherited Alzheimer Network, or the DIAN study for short. I'm not part of this study. This is a study that's being done in another laboratory. It's actually a network of centers around the world. And this is actually a pretty complicated slide, so I want to spend a minute on it. What these investigators in DIAN did is they assembled a whole bunch of people with Alzheimer's disease who have these dominantly inherited disorders, and they tested them for the gene, and they found people who didn't have symptoms, who had the gene but were preclinical. Some of these people were quite young. And one of the interesting things about these autosomal dominant Alzheimer's diseases is they tend to run true in families. So if your father got the disease when he was 45, there's a pretty good chance that you'll get the disease pretty close to that age, 45. I know, this is pretty depressing, it's pretty scary, but fortunately it's very rare. So what they could do is they could test people for all of these biomarkers and then figure out how soon or how long it would be before they got the disease. So they might test someone who was 25 who was destined or likely to get the disease when they were, say, 40, and they know that that person is 15 years before onset. So what they did is they measured all these biomarkers in people, and they put together this curve that sort of modeled how things were going to change over time, assuming that the data could be sort of extrapolated into this longitudinal data. And this is what you see. The very earliest things that are starting to change in these individuals are actually the amyloid, the A-beta. And it turns out one of the first things that starts to happen is the amyloid starts to go up in the PET scan. And that's here. This is the A-beta deposition. Now, I didn't tell you this, but it turns out you can also measure this A-beta in the spinal fluid. So you can do a spinal tap on someone and measure it as well. And in the spinal fluid, when it becomes abnormal, it doesn't go up, it actually begins to go down. And we think the reason it's going down is because it's getting stuck in the brain, so there's less of it in the spinal fluid. So at about the same time, the spinal fluid begins to go down, and the spinal fluid A-beta begins to go down, and the brain amyloid begins to go up. And it's only a bit later, actually, that you begin to see changes in tau, which we can measure in the spinal fluid. So that seems to be happening after the A-beta changes. And again, after all of that, the hippocampal volume begins to go down. That's quite a bit later. That's about 10 years later. So the CSF is starting to change 20, 15 years later. The amyloid, CSF, the amyloid in the brain, maybe 15 years. The tau, maybe 10 years. The hippocampus, about 10 years. And the glucose metabolism here also about 10 years beginning to go down. This test, this black line, is a cognitive test. It's a test that involves memory and judgment, orientation, and so forth. And that's the last thing to change. So that changes only very, very shortly before individuals would develop symptoms. So if you want to believe that these data represent the order of what's happening, what these data speak to is that the earliest changes are in amyloid, and later changes are in tau, brain atrophy, and glucose metabolism. And this, you can also look at this in sort of as a movie. So let me show you this movie. I'm going to show it to you a couple of times. Well, let me pause it for a second, because I want you to know what you're looking at. The top row is PIB, is amyloid, and as it goes from purple to red, there's more and more amyloid being deposited in the brain. These bottom scans are glucose metabolism and cortical thickness, and these started red when they're relatively normal, and as the glucose metabolism is going down, it's turning more blue. And as the cortex is becoming thinner, it's also turning more blue. So here we are now, about 20 years before onset. There's really no PIB in the brain. Maybe you can begin to see a little bit here. The cortex is not hypometabolic and maybe a tiny little bit thin, but let's watch what happens over time. More and more amyloid is being deposited on the top. Not much happening on the bottom. Still more amyloid. But now the glucose metabolism is really starting to go down. The cortex is starting to thin. It's sort of catching up. Now we're only 3 years before, and now we're right at the time when people are having symptoms. And it's obviously a process that continues. And in the late stages of the disease, there's amyloid everywhere. There's hypometabolism pretty much everywhere. There's cortical thinning pretty much everywhere. But you see, again, this interesting pattern that I showed you before with glucose metabolism. It particularly affects this medial part of the parietal lobe, the lateral temporal parietal lobe, and that's also where the amyloid is. And that's also where the cortical thinning is. I'm going to show you this one more time. Because now when you start to look at it, you're going to see that pattern begins to appear very, very early. And I'm going to pause it when it does. Okay, so now we're 8 years before onset, and you're beginning to see really substantial glucose metabolism going down in those same brain regions. The amyloid is already there. The glucose metabolism is starting to go down, and even the cortex is starting to thin 8 years before individuals would get symptoms. And as it goes on, 5 years, it's rather dramatic. So if this is a good model for now remember, this is autosomal dominant Alzheimer's disease. This isn't the typical type of Alzheimer's disease that older people get. But the idea is that this is a model for what's going on. And if it is a good model, then we know there are things going wrong in the brain 10 to 15 years at least before people get sick. So let's go back with those thoughts in mind and revisit some of these problems with the amyloid hypothesis. The first one is that there's a poor relationship between amyloid and cognition. Well, yeah, that's true, but it's not the amyloid. It's these other things that are the problem. The amyloid itself is causing these other problems, glucose metabolism, atrophy, and so forth, and the degree to which people have those problems reflects how sick they're going to be. So just to give you an idea of what these scans look like in normal older people, because that really is part of the story, this is a PIB scan in an Alzheimer patient. Again, this is from my lab. You've seen these before, similar ones just a couple of slides ago. And this is a normal older person, and this kind of light green background are areas of the brain where there's really no binding. It's just sticking kind of non-specifically. And this is an older person who has amyloid in their brain, who has a positive PIB scan. Now, I guarantee you that you could not tell the difference between these two normal older people. We give these people rather detailed cognitive tests. They spend hours in the lab taking memory tests, and there's virtually no difference in their performance. So they're doing just fine, but they have a lot of this amyloid in the brain. And if you want to look at this quantitatively, this is a graph. We actually studied some very young people in the lab here in green. These are people in their 20s and 30s. They have no amyloid in the brain, so you guys you're safe. The Alzheimer's patients are in blue, and our normals are in pink. And you see lots of these normals fall in the same range as Alzheimer's disease. They just have just as much amyloid in the brain as an Alzheimer patient. There's a bunch of them who have fairly low levels, and we think these are probably abnormal too, but lots of them have a lot of amyloid. And if you look across laboratories, about 30% of normal people in their 70s will have amyloid scans like this. so how do we explain this? Again, I'll go back to my model now, a little bit of an enhanced amyloid hypothesis, where the amyloid is there, but it triggers these other things. It triggers this neurodegeneration, which we think of tau pathology, dysfunction at the synapse, change in metabolism, brain atrophy, and it's that leads to cognitive decline and dementia. All right, older people have a lot of amyloid in the brain, but they don't have dementia. So how can that be? Again, it's the same basic idea, but now that we have these techniques, we can study older people in great detail. And in fact, there's a lot of data from my lab and other labs that suggest that these individuals, these normal older people, have changes in the brain that are very similar to the things we see in people who have Alzheimer's disease. So even though these people are normal and they're doing just fine, their brain is showing Alzheimer-like change. And let me show you some examples of this. So one of the ways we have been doing this is by studying networks in the brain, network connectivity. And I'm going to take you on a little side trip here about resting state connectivity or network studies with functional MRI. So if you go into an MRI scanner and you're not doing anything, and we're measuring the fluctuation in the what we call the bold signal, there's a spontaneous fluctuation in that signal that we think is due in part to neural activity. It's due to all kinds of things, but partially it's due to activity of your neurons. And these neurons are firing in systems, and in some of these systems the firing is synchronous. So one area of the brain will be firing in synchrony with another. And we can kind of drag this out of an MRI scan by any number of regions, any number of ways. And so, for example, suppose we decide we want to look at this region and that region. What we'll do is we'll sample those regions and we'll look at the time course of activity. And you see in this blue area, represented by this blue curve, and the yellow area, this is the spontaneous fluctuation of the MR signal. And they're relatively synchronous. They're happening at about the same time. Whereas when you look at the yellow and the green, they're asynchronous. So we would begin to infer that this blue and yellow regions together are functioning as a network because they're acting together. And there are many, many networks in the brain, and there are many different ways of examining these networks. But they're very robust, and they tend to reproduce from person to person and across different studies. And some of them are very simple. There's a motor network. So if you look at the motor area of the left hemisphere, you'll see that it fluctuates in synchrony with the motor area of the right hemisphere. But there are also cognitive networks. And one of the more interesting networks is called the default mode network. And this is what it looks like, extracted from the brains of a group of older individuals, where if you, for example, began to look in this brain region here in the precuneus posterior cingulate, you would find these other regions of the brain, the medial prefrontal cortex, the lateral parietal lobes, and the medial temporal lobes, were actually fluctuating in synchrony. And this is a very, very interesting network for two reasons. First of all, you probably, if you've been paying attention, realize that this has some similarity with the pattern of glucose metabolism change in Alzheimer's that I'm showing you, and the pattern of amyloid deposition. It's also an interesting network because this is a network that seems to be active when people are doing kinds of internal processes as opposed to external processes. So if I put you in an MRI scanner and ask you to do a task, actually, where you attend to a visual stimulus or an auditory stimulus, this network turns off. But if I ask you to remember something, it turns on. So it turns on during internally driven processes like memory. It also turns on when you're planning for the future. It turns on in a whole variety of internally driven processes, but turns off during externally driven processes. So it's also been called at some point, so it's called the default mode network because this is a network that seems to be the most active when you're not actually engaged in a clear outside externally driven task. So it's interesting for many reasons, and one of the reasons, of course, is because it seems to have very unique sets of cognitive functions. But it's also interesting because the pattern of amyloid very much replicates the pattern of this network. And it was not that long ago that someone made the observation that this network is falling apart in people with Alzheimer's disease. And it seems to be particularly affecting this network. So this is the default mode network in a group of healthy older people. This is the network in a group of people with Alzheimer's disease. And you can look at it and see that it doesn't look quite as healthy, not quite as robust, but you can actually do a statistical contrast between these two groups, and you begin to see these areas where the network is actually weaker in people with Alzheimer's disease than in the normals. And it involves these areas in the precuneus, in the parietal lobe, and actually also in the medial temporal lobe in the hippocampus. Now, as I showed you, this parallels the deposition of amyloid. So here's data from our lab where you see the default mode network mapped out in yellow, the amyloid mapped out in blue, and the overlap in red. And this red overlap is quite extensive. There are certainly plenty of areas of the brain where there's amyloid in blue but no default mode network, and there's some areas where there's default mode network and no amyloid, but there's a big overlap between amyloid and the default mode network. These are our healthy older subjects. So the obvious next question was, if you have more amyloid, is your default network in worse shape? And of course, I wouldn't be leading up to this if the answer wasn't yes. Here is a map of the default mode network correlated to the amount of amyloid in the brain. So these blue areas are areas where the more A beta there is, the less functional connectivity there is. So this network in these characteristic areas is beginning to come apart in people with Alzheimer's disease. There actually are some other areas that are sort of maybe increased in their connectivity. We're not sure what that means. It might be some kind of compensation. But the bottom line, the take-home point of all these the last 4 slides, is that this network is particularly affected by Alzheimer's disease. And we can see it beginning to become dysfunctional in normal older people who have amyloid in the brain. I think that's what the lawyers would call circumstantial evidence. So here's some more circumstantial evidence. This is the pattern of brain atrophy. And if we look at the difference between individuals who have amyloid in the brain and who don't, and sort of draw out the regions that are shrinking together, this is what we see. These are the areas of the brain that are shrinking as amyloid increases. Again, it looks very much like that default mode network. So again, this network is beginning to not only show disconnection, but it's also beginning to shrink. Again, maybe more circumstantial evidence. But here's evidence that I don't think is just circumstantial. These are our participants in our research project, tested for cognitive tests, and we kind of grouped them together in terms of memory function and memory function and non-memory function, and we separated them by their PIB status. These are individuals who have PIB positive or amyloid in the brain. These are PIB negative, and we compared their performance on these tasks. And as I was telling you before, you really can't tell them apart very well. There's so much overlap in these dots. You know, the amyloid doesn't seem to be affecting their memory or non-memory ability. They're scoring equivalently. But we're now following these. We've followed these people now over longer and longer periods of time, and we've now, at the point that this slide was made, we had about 4 years of follow-up, and the individuals with amyloid in the brain are beginning to show decline. Not all of them, but the mean has come down, and a few of the subjects seem to be doing worse. And so this, again, isn't Alzheimer's disease, but I think it's more than just circumstantial evidence. I think the idea that these individuals are beginning to show cognitive decline suggests that yes, indeed, there's something wrong. So again, I'll put this together in sort of this amyloid model just to look at it another way. It seems that, we have this beta amyloid in the brain, and some people seem to be able to tolerate it for a short time, maybe a very long time, maybe until they die. So there certainly are people who are going to have amyloid in the brain and will never get Alzheimer's disease. Other people clearly get Alzheimer's disease. And so what's the difference? Well, here we kind of invoke this idea of we call brain reserve, and we don't really know what that means. Many of us are trying to understand it better, but we think there's something about some of these people that makes them different. Maybe their brains are more plastic, so they can reorganize. I was listening to Alison Gopnik's lecture before, and I'm always reminded that things that happen during development are very, very important to things that happen at aging. So maybe their brains are developed in a way that their neural connections are more plastic or more resilient or more dense. And so when Alzheimer's disease begins to occur, they can withstand it for a longer period of time. Whatever it is, there seem to be some people who are somewhat protected and others who go on to develop tau pathology, neural dysfunction, brain atrophy, and we think these are the mediators of dementia. All right, let me end with the last challenge, which is the really important one. And that is that many clinical trials that have aimed at lowering brain A beta have failed. And these are in the news very often. They're very disappointing. Some of these approaches have used the kinds of ideas I mentioned, like immunotherapy, trying to get the amyloid out of the brain, blocking the aggregation into plaques, or blocking these enzymes that produce the A beta. So there's a number of conclusions you can draw from these failed clinical trials. One of them is that the amyloid hypothesis is wrong. The other is that waiting for symptoms of dementia is too late. So one thing I can tell you for sure is that we can lower amyloid in the brain. So this is one clinical trial of a drug called bapineuzumab that is an antibody to the A beta. And these are PET scans using PIB of individuals given the drug versus individuals on placebo. So here's one individual at baseline and then after treatment, and you can see that the amount of PIB binding in the brain has gone down. Here's another case, baseline, treatment, the PIB binding goes down with the drug, whereas the placebo cases are going up. And actually, the images are very nice to see, but these are the actual data where you see across the whole group of individuals in the trial, the placebo group went up a bit, the treated group went down. So this is all very consistent with the amyloid being cleared from the brain. But this trial and another one after it that was much bigger didn't produce any benefit to patients. So the operation was a success, but the patient died. So that's a big problem. What's wrong? Well, we can get rid of the amyloid, but is it too late? So one way to think about this is that this amyloid sort of initiates a process. The amyloid kind of goes on for a period of time, and it's aggregating, and after a certain period of time it triggers these changes in the synapses and in the neurons that eventually it sort of becomes uncoupled from the amount of amyloid in the brain. And so if we want to intervene, we have to think about when we intervene. If we want to affect the process at the bottom, we have to intervene at the bottom. So if we want to intervene at the top, we have to intervene at the top. So in some cases, if we're going to treat amyloid, we have to treat the amyloid before the disease is too progressed. Otherwise, we have to find other targets that affect the synapse or the neuron. So can we do that? Well, yeah. So now we can. So with all the stuff that I've been showing you, there actually are experiments that we can do to try to figure out if this approach is going to work. And the approaches use exactly the kinds of things that I've been talking to you about. So there are 3 clinical trials that are starting. 2 of them are beginning in groups of individuals who have these autosomal dominant genes. And they're going to be treated with these amyloid-lowering drugs. So we know we can lower amyloid. The question is, if we treat someone who has an amyloid overproducing gene, who has a mutation, if we treat them 15 years before they are due to get Alzheimer's disease, will we see changes in the rate of cognitive decline? Will we see changes in the other biomarkers? And so those clinical trials are beginning. You may have heard of one of these that's gotten a lot of press, that there's a family of a large kindred of individuals living in Colombia, South America, where many individuals have Alzheimer's disease. And these people are going to be treated with immunotherapy for Alzheimer's. And this other Diane study, individuals are going to be treated as well. And in a third study, they're actually going to be treating cognitively normal older people. And how are they going to get them? They're going to do PET scans. And as I told you, 30% of people in their 70s have this amyloid in their PET scan. So they're going to scan 3,000 people in the hopes of finding 1,000 who would then be treated with drug or placebo, again, to see if the outcomes of cognitive change over time can be affected. So, in my opinion, these are very important experiments. I don't know if the amyloid hypothesis is right, but I think many of us have been thinking for a long time that the only way we'll ever know if it's right or not is if we treat people and prevent Alzheimer's disease. So the bad news is that these are very, very complicated, very expensive experiments to do. But the good news is that they're doable. And if they work, they could have a major impact on the disease. So that's where we are. And this is kind of the way people are thinking about the way Alzheimer's disease, if everything goes well, this is the way the Alzheimer's story might end. With assessment of individuals' risk, a scan of some sort, some kind of selection of people for clinical trials, for example, who may be in what we will now call preclinical Alzheimer's disease, and then treatment with amyloid-lowering agents and the use of these drugs of the scans to actually measure the effect of the treatments themselves. So we're very hopeful that this is an approach that's going to work, but we have to wait and see. It'll be at least 5 years before we know. So with that, I will end. I want to thank all my lab people, especially all the postdocs, grad students, undergrads, and research assistants who do all the work. So thank you.
Time for questions on the panel later.
Okay.
Is that okay?
Yeah.